Good morning, and thank you for the opportunity. Today I will talk about a new smart region growing algorithm for segmenting single neurons from confocal datasets. The context is that of digitizing the brain at the microscale, and this is done for multiple purposes. First of all, the well-known connectome challenge, but also for many other relevant applications, such as the study of structure functions relations, as well as for documenting morphological alterations due to neuropathies. And to this aim, basically, we need optical access to neural tissues. And today we will focus on a well-established technology that is confocal microscopy, and on this new, recently developed clearing tissue technique, the Clarity, which allows to have transparent tissues, enabling light to penetrate, while preserving the original 3D environment of brain cells. And we will see that a standard segmentation algorithm may fail for this kind of data. So the reason of this region growing algorithm is basically to segment Clarity-treated tissues. You start standard by selecting a seed, and then a deep particle test is performed on the pixel distribution of a local crop centered on the seed to test unimodal against multimodal distributions. In case of multimodal distribution, the segmentation proceeds with the standard Otus method. Otherwise, a mixture model is fit on the data, since the foreground and the background may be mixed. The model uses a Gaussian distribution for describing the background statistics and the negative binomial distribution for describing the foreground. And basically, by tuning and controlling just the mixture parameter, we can set a soft threshold on what is foreground and what is background. And once this threshold is set, the segmentation proceeds on a local crop. New seeds are extracted from the local maxima of the distance transform, and then if we have new seed, the procedure is iterated, and otherwise we finish the segmentation. And what the SMRG basically gives you is a 3D smooth mesh of the segmented neuron that can be localized within the 3D environment of the brain. And here I report two case studies. First, validation on Clarity-treated tissues from mice brain. Basically, we evaluated the accuracy by segmenting these 3 neurons manually and with the algorithm, and we extracted the volume, the surface area, and the shell profiles of the segmented structures. And according to a Friedman test, we observed that there were no differences between the automatic segmentation and the manual one in terms of these morphological features. For reproducibility testing, we instead segmented 100 times each of these neurons by starting from different seeds and evaluated the coefficient of variation of these outcomes. And the maximum value we observed was around the 2.5%, suggesting the robustness of this algorithm to changes in initial conditions. Furthermore, we tried to compare the outcome of the SMRG with a standard algorithm available in the state of art, but many of them failed in the segmentation, so this first case study was performed only visually. A second case study is instead on a public benchmark, that is the olfactory projections fiber from the diamond challenge. And basically, we have the ground truth segmentations of these neurons. And we segmented each neuron with the SMRG and these 4 tools available in the state of art. And we compared each automatic outcome with the ground truth, but evaluating the internode distances. And in particular, extracting those distances above a certain threshold that allows to evaluate the substantial spatial distance and the percentage of substantial spatial distance, as suggested in this work. Just keep in mind that these are error values, so the lower the better. And what we observed was that the SMRG was comparable to these other tools available in the state of art for this public benchmark. So basically, I would like to conclude saying that it's not better for the standard benchmark is comparable, but at least when these other tools may fail, and this is the case of clarified tissues, SMRG can represent a valid alternative. Plus, it's easy to tune since we have just a mixer parameter to set up, and again, think about that mixer parameter as a soft threshold. I have to say that this is not comparable yet in terms of computational times, since the procedure is quite slow. So we need to speed up and to further test on other published benchmarks. If you would like to contribute, you find the code on GitHub. And so thank you for your attention, and thanks to the Flagira who recently funded us for keeping working on this. Thank you.